Yeah, we have seen the biometrical techniques we used for assessing the variability, and now we are discussing about how we can able to select better individuals by using biometrical techniques. We employ the different mating designs to understand, to estimate the different components of variances, GCA, SCA. Okay, and we have discussed in detail GCA, we have discussed in detail about SCA, how to estimate from different cross combinations. What is PCA? We have estimated. What is discriminant function analysis? What is cluster analysis? What is D-square analysis? What is metrograph analysis? Okay. And now we are going to discuss about what is mating designs and what are the different mating designs at present and how we can use in the plant breeding. Then why we need, what is mating design and why we need it? Mating design means what is mating designs means various cross plants or is a system to develop progeny and evaluation of strain uh, in the terms of the genetic background is known as mating design means we are planning the crosses. For example, either you cross with parent 1 and parent 2 and F1 and F2. You select randomly from F2, then again you analyze it, or you can make the different crosses and you create the progenies from the crosses and evaluate their progeny. Means the controlled crossing in a definite fashion and the creating progeny and evaluating the progeny and we are estimating the desired estimate. This is what the mating design means. Means why we are creating mating design to test the significance of genetic component and estimation of genetic component. First, we will estimate and we will test for significance. Then, Coker-Hamann in 1963 classified the mating designs based on the partition of the variants and the parents. There is different classification of mating designs: one factor designs, two factor designs, three factor designs, and four factor designs. In one factor design, the variance is just genotype partitioned into genotypic variance and environmental variance. Because we have only one type of variance we can be estimated from this mating designs that are like polycross designs, offset family designs, and top cross designs, where only additive variance is estimated, but other we can't estimate. That's why it is only one factor designs. Another one is called as two factor designs, where we can partition the total genetic variance into additive and non-additive components. The example for these two-factor designs, they are called as diallele designs and your partial diallele, line tester, and North Carolina designs. Then another one is three-factor designs, where the genetic components is splitted into interactions, epistasis, and this can be estimated into in trial crosses or triple test cross designs. And another one is four factor designs. We can have A plus D plus I, and we can also estimate the order effect. Means what is the parental control of the progeny that can be estimated in quadriallele and double top cross test analysis. Then why we need to like what are the genetic estimate that we can get from different mating designs we can observe, like in a top cross or a polycross we will get the variance due to only females and we can estimate covariance of opposites. Then we can estimate the parameter like 1 by 4th of genetic additive variance. Then means covariance of opposite is equal to 1 by 4th of additive variance, that is equal to your variance of your female. If it is a line into tester or NCD3, the variance due to males is equal to variance due to female, that is equal to variance due to your GCA. Then it is includes covariance of opposites. It is like 1 plus F. F we discussed like inbreeding. It also takes the account of inbreeding coefficient that we have discussed in the last recorded class, the last 9 to 10 class. Then of additive variance. We will discuss one by one, like what is this and what are the parameters that we are estimating in which fashion we have to make the crosses in the next coming slides. First design was, as I told, is the first degree designs, the top cross and polycross designs. It actually developed by scientist Davis. It is used for in testing of our inbred lines, which is the better inbred based on the what is top cross means your crossing inbred into any open pollinated variety. Means a broad genetic base open pollinated variety so that we are getting progeny. We are evaluating progeny then to assess the worth of the inbred. Okay, here see, and when you are crossing inbred with the OPV, the progenies are half sips only. Means only one parent is common, but rest other parents are random from this broad genetic based lines. Then there are full all are hop sips only, and you are using only single wide tester. Okay, we having single broad genetic based tester. Then we can use ANOVA. We can split the variance components. Okay, then we can assess. For example, here the variance covariance due to half sip is equal to 1 by 4th of dominance variance and 1 by 4th of additive variance and 1 by 6th of your additive into additive variance. Here we can't estimate the dominance variance because here we are not creating any full sips. Okay, because of only half sips, we can able to assess either additive or additive into additive interaction only. That's why discard. We can partition the variance into only one component, that is additive component and phenotypic component. That's why it is called as single factor designs. Then another one is diallele cross analysis. What exactly this diallele cross analysis means? Diallele cross analysis means we are mating the selected parents in all possible combination and evaluation of set of diallele crosses in a known diallele analysis. Means diallele mating design is used to evaluate several inbred lines in terms of combining ability and variance and effects. And the dial mating design involves three types of materials. One is we involve in direct crosses or one-way crosses or reciprocal crosses or the parents that we will discuss. For example, we having set of inbred 1, inbred 2, inbred 3 and inbred 4. We are making crosses, we are making the inbred 1, inbred 2, inbred 3 and inbred 4. We are crossing inbred 1 with all possible combinations. Then we are creating F1s. And you are evaluating F1 for assessing your GCA and SCA or any variance components. There are two approaches for this diagonal analysis. One is Hyman's graphical approach or graphical approach or Griffin numerical approach. Hammond's graphical approach means that this is a graphical approach which involves construction of VR variance and covariance graph, VR and WR graph, analysis based on the estimation of component variation. We can estimate additive variance, dominance variance, H2, E, F, and H2. Here D is additive genetic variance, H1 is dominance variance, where H2 is the H1 into 1 minus U minus square. Actually, it actually tells about this H2 component will tells about the symmetry of your the genes. Then another components are U and V. They explains the proportion of positive allele and proportion of negative alleles in the parents. Then E is expected environmental component and F means F are over the array. Actually, I'm not going in detail because this is very difficult to understand also. That's why I'm giving the basic fundamental things of every mating designs, and in exam this is only going to ask. Okay, S square is dominance effect. Then there is a, how to visualize this one. Okay, WR is covariance and VR is variance. And many times asked, WR is placed in which axis? WR placed in Y axis and VR is placed in the X axis. This is very, very important. Many times to ask the exam. For example, this array is splitting the WR axis. It means that there is no dominance. If it is splitting the y-axis but touching the limiting parabola, we can say there is no dominance. If it is splitting the y-axis but away from the limiting parabola, then we can say that there is an existence of partial dominance. If this array is split exactly into the origin, then we can say that completely dominance. Then the array we split the x-axis and the value is negative, then it's called as over dominance. And analyzing this very, very important. Many questions were asked in the exam. For example, when a regression line passes through the origin, it indicates complete dominance. You can see this regression lines, it passes through origin, it is a complete dominance. It is right. It is true. When the regression line passes above the origin, Cutting the WR axis, it suggested absence of dominance. See, touching the parabola and cutting the WR axis. See, when the regression line passes through above origin, cutting the WR axis suggests the absence of dominance. No, it has to touch the limiting parabola. Then, when the regression line, it is false actually. Then, when regression line passes above the origin, cutting the WR axis and touching the limiting parabola, it suggests partial dominance. No, it is no dominance. It is also false. Then, when the regression line passes below the origin, cutting the VR axis, it denotes the presence of over dominance. You can see when the regression line passes between cutting the VR axis and negative. Below the x axis, it is called as over dominance. It is correct. Only A and D is correct. That's why A and D is the right answer, option 3. Okay. Then another method of diagonal analysis is Griffin numerical approaches. Means we are making it is based on the estimates of combining ability variance. See that the additive variance is equal to 2 times of GCA and dominance variance is equal to variance of SCA. And you should know what the GCA variance and SCA variance in the last class I have discussed in detail. Okay, that's why I'm not repeating here. Then what is this? Here we having partial diagonal and complete diagonal. Okay, when we include the reciprocals, it is complete diagonal. When we are not including the reciprocal classes, it is called as partial diagonal. You can see that based on the n is equal to number of parents, where the straight crosses are includes number of n into n minus 1 2. When we include reciprocal crosses n into n minus 1 by 2, and the total is n square. This is one diagonal, and then the second diagonal, the total number of crosses is equal to n into n plus 1 by 2. Here, we are excluding the reciprocal classes. We are including only straight crosses and number of parents. Okay, but in this case, you can see only straight crosses are included with the parents is only n into n minus 1. If you are only n into n plus 1 divided by 2, it is you are avoiding your reciprocal crosses. Okay, it's normally most of the questions are from how many you having this many number of inbreds, like you having 20 number of inbreds in a partial diagonal. How many number of crosses you are going to get in a diagonal fashion? That you should able to answer. Then many times asked in the exam. Means your partial diagonal design is, then another design is called as partial diagonal design. It is actually proposed by Campethron in 1957. It was first elaborated by Campethron and Cournot in 1961. In partial diagonal, where the total number of crosses is equal to ns by 2, where s is the sample numbers. Okay, s should always greater than or equal to n by 2. For example, you having 100 n, then you just divide it by 2, you'll get 50. Means either 51 you can use. Okay. Then purpose of sampling, the constant k is worked out as k is equal to n plus 1 minus s by 2. For example, how the design looks like. For example, this you take the inbreds 1, 2, 3, 4. Here 1, 2, 3, 4. For example, your k value is, sorry. For example, you are making 1, 2, 3, 4. 1, 2, 3, 4. For example, your k value is 3. Means you start selecting the individual from the third. Okay. This is what your partial diagonal is. Means you are only analyzing some set of crosses from the total set of crosses. We are not evaluating all the crosses. It's a very time consuming. That's why only some set of crosses are evaluated. That's why it is called as partial diagonals. And here the total number of crosses is ns by 2, where s is you can use n by 2 formula. Then for sampling purpose, we have to use the formula n plus 1 minus s by 2. Here we can estimate both SCA variance and GCA variance, but we can't estimate SCA effects. Then another important design is called as line into tester cross design. Line into tester cross design means it's a basically extension of top cross that we have discussed. Here number of crosses is equal to number of males into number of females. For example, you have 10 males and 5 females. Means number of crosses is 50. Like this, we are estimating the number of classes by using number of males and number of females. And it is also regarded as half SIB and full SIB analysis because in this design we are creating both half SIBs and also full SIBs. Here we can use many testers. At least three testers we have to use. This year the question was asked how many tester we had, how many minimum testers we have to use in the linearity tester. At least two also we have to use. If you use one tester, what will happen? For example, you have to analyze this data with ANOVA only. For example, you're using only one tester, the degrees of freedom will become zero. Then how do you estimate? That's the minimum two tester is required for the analysis of linearity tester design. Then here covariance of offset is equal to variance to GCA and variance additive variance is equal to variance due to GCA. Your dominance variance is equal to variance due to your SCA. Your covariance of SCA is equal to covariance of full SIPs minus 2 into covariance of half SIPs. What are the different characters of testers? Here, tester should be broad genetic base and is able to differentiate genetic worth of individual. For example, you are selecting four individuals. Two tester. For example, your tester can able to discriminate these individuals. Means it able to assess the genetic worth of these individuals and should after selection. For example, if selecting this particular individual based on using these testers, you should maximize the genetic gains. Means you should improve this particular inbred based on this tester. And the tester should have the broad genetic base and you should clearly classify the performance of the lines. Means you should be able to discriminate the individual. And also along with that, it should be easy to use. Means simple to use in the design. Okay. These were the characters of the testers. Then another mating design is called as biparental mating designs. Means we are crossing a randomly selected plant. For example, you create P1 and P2. It's also called as biparental designs or North Carolina designs. You are creating F1 and F2. Then you select random samples from F2 and making the crosses with both the parents. They are North Carolina designs. How you make the crosses, it depends on different designs called as North Carolina Design 1, North Carolina Design 2, North Carolina Design 3. Like, which among the following is not one of the assumption of biparental cross analysis? As I discussed in the Hardy-Weinberg assumptions, if you are listened, if you are or watched my that recording, you can able to analyze this. Means absence of multiple exactly and absence of epistatus exactly, absence of regular diploid. Okay, not absent. It should presence of regular diploids. We avoid that if any polyploid segregation is there, it will wrongly estimate your all additive variance or dominance variance or genetic components if it is a polyploid segregation. That's why we assume that this is following diploid segregation. There is no multiple alleles, there is no epistasis, there is no maternal effect. Then when it comes to North Carolina Design 1, it's a polygamous design where one male is crossed with more than one female, chosen randomly, and each male is different groups. For example, here we create the sets. For example, you're having 100 F2. You randomly select 4 F2. Then you can make crosses among them, each one. Then you can create sets. Some you regarded as male and some regarded as female. Okay, here there is no, for example, you're having 100 F2. So 4 is regarded as male and you make crosses, you can create 4 individual, you can create one set. Another you regarded as a male, created crossed with another set of females, it is another set. Here N is number of male you are designating and S is number of set. The total number of crosses is N into S. Here the variance created due to males is of 1 by 4th of additive variance and variance created due to female is 1 by 4th of additive variance plus 1 by 4th of dominance variance. It influenced by the presence of maternal effect. If there is any, we have discussed in detail what is maternal effect, what is the genetic phenomenon of maternal effect. Please watch my recording on maternal effect. Okay. Then if there is a maternal effect, then definitely we are regarding some set of individuals as a female. If there is a maternal effect, what will happen? You are going to get, they are having all the character from the your female set only. That's why we should assume that there should be there is no maternal effect and it is also influenced by the maternal effect. Then Another design is called as North Carolina designs. It is also called as factorial designs. It is also influenced by maternal effect. Here, what we are making, it is just some lines are regarded male, some regarded like in F2 population, some males are regarded as males, and some individuals regarded as females, and making crosses among them. It just look like your linearly tested design only. It is also called as factorial designs. Here the number of crosses is equal to m into ns. Okay, here the variants are partitioned into variance due to males, variance to females, and variance to male into females. Here variance to male is 1 by 4th of additive variance, variance to females is 1 by 4th of additive variance, but variance to male into females is 1 by 4th of dominance variance. Then another design is called as North Carolina designs. Here something is different like P1 and P2. We are developing an F1 and we have an F2. Here F2 is crossed among them along with P1 and P2. Here, the total number of processes 2 into ns, and variance can be partitioned into one. Here, variance due to males and variance due to male into females. Variance due to males is equal to 1 by 4th of additive variance, but variance due to male into females is called as 1 by 4th of your dominance variance. But one of the greatest advantage of North Carolina Design 3 is it is not influenced by the presence of maternal effect. This is one of the greatest advantage over both the methods. And here, estimation of linkage is dissipated by the backcrossing in NCD3. Therefore, because we are selecting F2 and we making the backcrosses with the parents. That's why it is your linkage is dissipated. We are breaking the linkage because where estimates are, if any linkage is present in F2, it will make non-segregation, like independent, non-dependent segregation will be there. To avoid that effect, we are making this back crosses. Okay. Then the order of precision when we are estimating NCD3, you are estimating the genetic components through NCD3 having maximum precision over the NCD2 and NCD1. And NCD3 will require very minimal area when compared to NCD2 and NCD1. You can solve this question with the minimum available resources. Which mating design should be used for biometrical cross analysis? NCD3 will be the best answer. Then, which of the following mating designs involves the selection of several random parents P1 and P2 and F1 and F2, selection of several F2 plants and from the F2 plants, and they are back crossing to the males each of the parents. We are back crossing to the each of the parents. In which design we have discussed NCD3. Okay. Then another one matching design is called as triple test cross analysis. It's just an extension of NCD3. Here what will happen? Your parent 1 and parent 2 is crossed. Your F1 is created and F2 is created. This F2 is backcrossed to P1 and backcrossed to P2 and is also backcrossed to F1. This is the only difference between your NCD3 and NCD TTC. And each randomly F2 plant is crossed with inbred parents along with F1, and we require 4 cropping seasons to do this evaluation, and it tests the presence of epistasis or not. Rest all other designs we have discussed now, it is no test will decide estimate the epistasis. And here it just present, it's not quantify the epistasis, but it just it says that either epistasis is present or not. The epistasis that can assume by L1 plus L2 minus 2L3. If it is summation is equal to 0, then we can see the absence of epistasis. Then this is about the mating designs. Then another one of the important analysis is called as generation mean analysis. Okay, generation mean analysis means this concept was actually developed by Hammond and Jinks and Jones in 1958 to estimation of genetic components of the variation. Here, what will happen? We have different models of generation mean analysis. It is completely based on the mean, means first order statistics, which is having more precision. And here, what we'll do first, we will do scaling test or joint scaling test. Then we will see that presence or absence of epistasis. Based on that, we have a different models like 3-parameter model, 3-parameter model, 5-parameter model, 6-parameter model. Then we are analyzing the different genetic components of variance. Then Actually, I'm going first because nobody is there to ask the doubts. Okay, normally I expect the doubts because it's very difficult. Okay, biometrical genetics is very, very, very difficult. If you are reading the book, you will not go into anything. Okay, but unfortunately, no one in the class. That's why I'm covering. Any doubts, like you can text me, but I can't repeat these things again because it's very hectic to do this again. Then the significance of A, like first we will see what are the different models of GCM. We have six parameter model, five parameter model, three parameter model. In six parameter model, we can estimate uh, all the parameters. Like we have estimate additive variance, dominance variance, and epistatic variance. Okay, we can using population used in six parameter model. Parent one, parent two, F1, F2, backcross one, backcross two. In five parameter model, we can estimate only MDH, because, but additive into dominance we can't estimate. And here the population used are parent 1, parent 2, F1, F2 and F3. And here no J means we are not estimating additive into dominance variance. In three parameter model, we can estimate mean, the additive variance, dominance variance, but we can't estimate any epistaticity. See, all your I, J, I, L, it's not estimated in three parameter model, but we are using all the six population. Generally, students will confuse three parameter means we are using three set of population like parent 1, parent 2, F1. It is not like that. We are using all the six population, but we are estimating only three parameters like additive, dominance, and mean. But here in five parameter, we are estimating mean, additive dominance, but only one type of epistatus is not estimated, that is called as additive into dominance. Here, instead of backcrosses, we are using another one is F3. But in six parameter model, we are having backcross 1, backcross 2, and F1, F2, P1, P2, and we are estimating all six parameters mean, additive variance, dominance variance, and all the types of epistatus. That is the only difference between these models. Before going to use this model, we having important test is called as scaling test. Scaling test which are given by math. Actually, what are the different scales we can observe? A scale, B scale, C scale, and D scale. What exactly means? If there is a significance of A and B scales, it indicates that there is a presence of all the three types of non-allelic gene interaction, like additive into dominance, or dominance into dominance. If there is only C scale is significant, we can suggest that only the dominance and dominance type of epistatus is present. If the significance of D scale, it only tells about there is a presence of additive type of gene interaction. And if the significance of both C and D is there, additive and dominant type of interaction is present. Please remember, when A is significant, what is then? When A, B or both are significant, what is the phenomena? When only C is significant, what is the phenomena? When C and D is significant, what is the phenomena? What is the D scale is significant, what is the phenomena? That you should know. Then this is about the models of GMA and the different scaling tests. And another one is nowadays we are using joint scaling test. It is proposed by Cavalli, sorry, scientist Cavalli. Here what happens, here we can able to estimate only one scale at a time. We can't estimate all the scale together. That's why in giant scaling test, we estimate the mean and we calculate the expected mean and observed mean. We'll do chi-square test for expected mean and observed mean. Based on the significance of chi-square test, we can estimate all the genetic parameter and we can say that, okay, there is a presence of epistasis or not. And that much detail is not needed. What are the information present in Kundan Singh and SSRI, that is more than enough for the ASRB net. Then another thing is gene into E interaction analysis. Okay. We have time and we'll discuss. These are all the things are very, very important. I thought it will go for many classes, but fortunately it went very fast because no one is there. Then GE analysis means we believe that P is equal to G plus E. In all the estimates, we can see all the quantitative phenomenon that I had told. It's only based on the assumption of P is equal to G plus E. But when the same individual subjected to more than one kind of environment, the P is the phenotype is more than your genotype plus environment. Sorry, genotype plus environment. Why this phenomenon occurs? Okay, it is not just a summation of G plus E, but it is more than that. What is that? That is called as G into E interaction. Okay, G into interaction. Now, means whenever the individual, for example, if the A individual grown in a different environmental, it will not give same performance across the environment. It will have the differential performance in the different environment. This is called as G into E interaction. We should properly estimate the G into interaction. Then only we can able to properly estimate all the genetic parameters. Then then before going to understand what is G into E interaction, we must understand what is environment. Environment, there are two types of environment, microenvironment and microenvironment. And first we'll discuss about microenvironment. Means, for example, you're growing your plants in the field. Okay. Each individual of a population subject to specific environment of its own. See, the surrounding environment in a single field itself is called as microenvironment. Either how much radiation this plant is getting, how much disease pest it is getting, what is the soil factor around this plant is microenvironment, what are the weather fluctuation around this plant is well microenvironment. And it is particular period, it confined to particular period of time and particular site of the particular individual is present. And actually, this classification was given by Comstock and Moll in 1963. Then another type is macro environment. Means, for example, latitude or longitude of the particular area, like it's a hilly area or plot area, or it's a semi-arid condition, larger scale environment. Okay. Then there are predictable and unpredictable components also we can classify. Predictable means permanent attributes of environment. Okay, it's not going to change. But unpredictable, sudden drought will happen, sudden flood will happen. They're all unpredictable component of environment. Then what is G and to E interaction? Means the phenotypic performance of a genotype is not necessarily the same under different agroclimatic conditions. Means all the genotypes may not reach the same level of phenotypic expression under all environmental conditions. For example, if the genotype is grown in a multiple environment, this genotype doesn't completely express its phenotypic expression in samely in the all the environment. Means there is a G into interaction. We need to estimate it. How we can estimate? Before going to that, we have to analyze what are the different types of G interactions are possible. You can see in the first diagram. See, these are the different inbred lines. Okay, these are the performance. Okay, then you can see the individual A in the environment one. This is the X environment and this is Y environment. See, in X environment, the individual A is having low performance. When it is shifted to Y, it is having high performance. Same like inbred B, the variety B, it having low performance in Y environment, so X environment, it is high performance in Y. It is means a linear G interaction. Means the variance is very same. Like rank of the genotype is not changing here. Okay. This is also called as linear gene to interaction. But you can see here, this is called as crossover gene interaction. Here, rank of the genotypes is changing. See, in A, in X environment is giving very nice, and in Y environment is giving very bad. But you can see the B, in X environment is very bad, but in Y environment is very high. This is called as cross GE interaction, where rank of the genotypes are different changed. Here you can see, here both are giving equal, and here both are opposite. Here both are crossover. Like this, here both are high environment in the both are giving same yield in Y environment, but both are having different yield in X environment. Based on this, there are n number of types of classification we can make, but these six type of environmental interaction are very important. Then why we need to study this GE interaction? Because if we, first it interaction distorts the genetic analysis. How? because the absence of proper estimate of G is confounded with the G less to overestimation of the G. Okay, P is equal to G plus E. If we are not taking G and E into componenter, we are estimating both the G and both the E. These overestimates are not useful for the plan bidding. That's why we need to eliminate this compound, or either we have to consider this compound. Okay, if you are neglecting this compound, your estimates will going to wrong. Then misinterpretation of the results or selection response is altered or various miscalculations like we can miscalculate heritability values or the genotypic slippage occurs. Anything we can happens if we are not considering G into E interaction. And it's very difficult concepts, but no one is there. That's why I'm going very speed. I can't explain myself. Okay, I studied myself. Some doubts are coming, then only it's very easy to explain. Okay, myself has to question, myself has to teach. It's very difficult task. Then means what happens? GE interaction it limits the adaptive range of an individual, and genotypic stability over the time and space depends the minimizing the impact of G interaction. Then we had to choose the breeding methods based on the G interaction. We can select, we can breed the genotypes for the specific environment, or we can breed the genotypes for the broader environment based on the type of gene interaction present in the genotypes. Either we can having two strategies, narrow adoption strategy or broad adoption strategies. Then how we can estimate this? Okay, first we can estimate G interaction either in three ways. Either you can go with G into location basis, or the G into year basis, or we can go into G into location plus year. Multi-year, multi-location, or multi-location, multi-year, combinedly we can also do. Based on this, we can estimate your GE interaction. How we can estimate that we will discuss. Okay. For example, we have larger GY variance means it requires testing over multiple years. If large GL where it requires multiple years, multiple locations, we need to evaluate for the GE interaction for estimation of GE interaction. Then how to analyze the GE interaction? Either we can use conventional methods like stability factor model which proposed by Lewis in 1954 or equivalence model by Vrickel in 1964. Either you can use stability variance model by Shukla or we can use Lin and Bin's model in 1988 and descriptive method of grouping phenotype by Francis and Korenberg. I'm not going to discuss all those things, but all those things are not important. But stability factor and equivalence model are very, very important. Then either we can analyze the G interaction by classical ANOVA. You can split the variance component into G into location, G into year, or location into year. Then this is first order. In second order, we can combine all the things G, L, Y. Then we can analyze, we can split the variance components. Okay, how much variance is due to G into L, how much variance is G into Y, how much variance is to L into Y, we can estimate. Based on that, we can estimate the amount of G interaction is there in the population. Either we can use correlation models, either we can larger correlation between the G and GE, then it shows that high G interaction. Based on that, we can able to study. And another based called as regression coefficient models. It is very, very important. Still now, where many are using regression-based model. One model is called as Finlay-Wilkinson and Eberhardt and Russell model, Perkinson and Jinks model, and Freeman and Perkinson model, and genotypic stability model. Then another model is called as principal component analysis. Also, we can use or shifted multiplicative model, redundancy analysis, factorial regression model, GGE biplot model. Anyone we can use. Then like one I told like stability model and equivalence model are very important. Means stability means it actually given by the Lewis. We had to estimate stability index. Means what is stability index means the mean of the individuals in high environment divided by mean of the individual by low environment. Based on that, we'll estimate the stability factor, and this stability factor used to interpret your GE interaction. Another one model is called as equivalence of the genotypes, which developed by Brick. It means that contribution of the genotype to GE interaction is the sum of the square of the express of this percentage. Lower will be the equivalence, higher will be the stability. Then the another regression based model is called as Finlay-Wilkinson model. In 1963, he explained this model. Here, based on the regression coefficient, we can classify the stability of the individual. If the regression coefficient is 0, we can say that the particular individual having absolute stability and well adapted to all the environment. If the regression coefficient is 1, we can say that it having average stability and it's well adapted to all the environment. Then if it is regression coefficient is more than 1, we can say that the below average stability and specifically adapted to favorable environment. If it is regression coefficient is less than 1, it is above average stability and specifically adapted to unfavorable and poor environment. And in every analysis we are had to do ANOVA. You can see this ANOVA. Here we can partition the variance into genotype, environment, genotype into environment, regression, and the deviation from the regression, and this deviation from the regression is very important. Okay, thus deviation of regression only the regression coefficient, based on that we are classifying this one. Then another, nowadays everyone using all these are nobody is using, don't worry, everyone we are using, even I am using for my PhD for stability analysis. They are called as AMI and GGE biplot analysis, GGE biplot analysis. What is the SUMMI model? It is the combination of both principal component analysis and ANOVA. Means ANOVA is regarded as an additive main effect, and where PCA is made as PCA, interaction effect, multiplicative interaction effect. Here, I, if you know the PCA, then you can able to understand this. I have discussed PCA. If you listen this class, you can easily interpret this one. Here, ANOVA, what is the function? Why we need to do ANOVA here? Here, estimate the genotypic deviation. Like ANOVA means here only. How much genotypic deviation? How much GE interaction? We are clubbing this result with PC analysis. What PC analysis will do? It will create the scores. Which interaction having maximum variation? Thus, clubbing this regarded as Hummy model. Then why we need Hummy? Why it is called as hybrid model? It means that it uses both additive effect from ANOVA and multiplicative interaction from the PCA. And it provides least square analysis for better interpretation. And we can visualize in biplots, okay, based on the vector. Here some genotypes are there. This is one vector, this is environmental one. How many genotypes are performing better in environmental one, that we can estimate from AMI model. This is means when to use the AMI: when the data is structured in two-way factorial designs and required three rows and three columns with replications, then it should be quantity to trade, and when effective main effects and interactions are significant. First you do ANOVA, then this should be, we will get significant effect, then only we can use your AMI model. Then another important is called as biplot model. Here GGE, gene and gene interaction, GEE interaction biplot model. Here here, GGE biplot means here we are ranking the genotypes based on the interaction with the different environment. We can identify the best genotype for each environment by one graph is called as which own where pattern environment. And you can able to define which is the mega environment. Means which environment will support higher expression of the all the genotypes. You will see in detail. See, for example, this is the typical graph of which own where graph. Means in which environment, which genotype is performing better. See here, the environments are classified, and they are here. See in environment 7 and environment 5, G3, G11, G5, G2, G18, G6 are performing better. And in environmental 8, and you can see some genotypes are extreme to the environment. See, like based on that, we can interpret which genotypes are performing in which environment and which is the better environment. See, this environment, this is called as mega environment. Most of the genotypes are performing the better here. Like this, there are many interpretation we can draw from GGE plot, but GGE by plot. Then, purpose of GGE plot and AMI analysis, both the methods used to analyze multi-environmental trial data to evaluate the crop performance. and it combines both G and GE interaction, whereas AMI analysis separates them. Okay, then we can compare the GE biplot AMI analysis. Which one to use? For example, you take for the mega environment analysis, I prefer that GGE biplot is very good. It's based because it can able to identify which own where pattern so that we can easily identify in which environment having maximum genotypes are having good. But in AMI analysis graph, we can't estimate it easily. Then, for example, if you want to study genotypic evaluation, in GGE plots, mean versus stability we can see. We can view what is the mean value of the genotype and how much stability of the genotype in the graph. But in the AMI plot, it is an inner product property. It is visualization is very difficult. And if you want to analyze the test environment, GG plot distinguish between discriminating and representative environment. It can create the representative environment, and which environment is best to test your genotype can analyze from the your GG plot. But from MA analysis, we can't do that. It's very difficult to do that. That's why the key advantage of GG plot, it is more explanatory power, more visualization, and it can easily select the superior genotypes for the based on the environmental graph. But criticism for AMI analysis, it do not efficiently handle complex gene to E interaction. That's why GGE biplots are more better than AMI biplots. Okay, then we can solve some questions about what we have discussed based on the quantitative genetics. Okay, I expected a lot of interaction from these topics because while reading myself, I got a lot of doubts by checking in the internet, by asking my seniors, I got clarified. But unfortunately, here no one in the class. I'm covering without interaction. It is really unfortunate. Okay. Then, for example, if you take the GMA analysis, there is one question asked in 2023. Three parameter model, the generation mean analysis P1, P2, F1, B1, B2 is used. See, three parameter. But I told like six population we need. Here only one, two, three, four, five population is there. Statement one is wrong. Okay. Then the model does not provide the information about episode. Absolutely true. Here statement one is wrong. Statement one is correct. Then here one question asked in the 2024. See, which one where view of the GG biplot is superior for AMI analysis that we have discussed for the mega environment analysis, where GG biplot explain more G into G than AMI one graph and it's easier to construct and visualize. Both are correct, okay, and R is the correct explanation of A. This is very, very important that we have discussed while comparing GG biplot and AMI biplot. Then in a population of maintained by random mating, five pair of parents in every generation, what will be the inbreeding coefficient after second generation? Please solve it. I have given the formula in last classes. Okay, what is the formula? Here I will write. For example, we having the five pair of the genes, and I will write solve by pen so that it will be easy. Like five pair, we have five pair of genes. We have five pair of genes. And in every generation, what will be the inbreeding coefficient after second generation? You just simply substitute this five pair of parents. See, we're having effective population 10. Five pair means 10 individuals are there. And they are maintained in second generation. What will happen? You can use the formula which I have discussed in inbreeding coefficient. Oh, we covered this much. So this formula, you just substitute 1 minus ft minus 1. We have ft minus 1. 1 minus 1 by 2 power over 10 means 20, and we have to estimate only 2 second generation. Okay, then if we substitute 10 here and inbreeding coefficient of the generation, what is the inbreeding coefficient of the generation? You can substitute here, we will get the uh, inbreeding coefficient. Okay, you substitute and give the answer to me. Okay, I will discuss. Then what is happening? Then it is actually to genomic selection. I will discuss after discussing genomic selection. This is for statistics. Normally, well, after doing ANOVA, we will go for significant the grouping test, LSD or DMR. We know what is least square designs and Duncan multiple range test. Actually, we know that when we have unplanned comparison, it's better to use DMRT. When we have planned comparison, it's better to use LSD. It's basic statistics. Okay, both statements are wrong. Then we have discussed the line entry tester analysis. Here, the minimum two testers are required for a line to analysis. Exactly true. Okay. If you are using one tester, your degrees of freedom will become zero. It is very difficult to analyze. Then tester should be related to the pedigree. See, if it is related to pedigree, your overall estimate will become wrong. It should be as diverse as for the from the inbred parent. That's why it is wrong. Then tester should represent the broad genetic piece exactly. 1 and 3 is correct and 2 is wrong. Okay. Statement 1 and 2 are two. Statement 1 and statement 2 are no. Statement 1 and 3 are true and statement 2 is false. This one is right. Then, if an inbred is crossed to develop an hybrid produced through double haploid technique, the difference in the gene frequency Y either 0, either 1. We have discussed double haploid techniques because it's a pure inbred. Either one in one pair, one inbred having 0 and another inbred will get 1. This is clear. Then, if a two inbred line is single cross, the differ for k number of loci. In case of extreme dispersion, what will be the positive values that are increasing the value of the phenotype or presenting one inbred line? Then coefficient of dispersion. See, it's saying that if the two inbred lines of a single cross hybrid differ with k number of loci, and in case of extreme dispersion, always remember in case of extreme dispersion, where RD is equal to 1. Okay. For example, all the positive alleles in one parent, all the negative allele in one parent, it is called as extreme dispersion. Whenever the extreme dispersion, the coefficient of dispersion will be 1. I will discuss these things in biometry of heterosis. I think I will take tomorrow. Okay. Then the selection is most effective when the gene frequencies are definitely when q is equal to 0.5. Okay, we have, and when the p is equal to q is equal to 0.5, selection is maximum selection response that we have discussed. And when we have q is equal to 0.01, when selection is very least effective. Then, which of the following multivariate analysis allow interdependence model? As I told, PCA is interdependence model. We just check correlation or discriminant or canonical. All are based on correlation. Means one variable is dependent on another variable to estimate the overall effect. But in principal component, it takes the as independent variable. That's why it is called as interdependence model. Then, when allele A mutates to the frequency of 1, 1 in 10,000, and it back mutate when 1 in 10, 1 in 10,000. Just to forget about all this one, you just check it say the population in equilibrium. You should know that p square plus 2pq plus q square is equal to 1. This should be there. Whatever the question, if we say that the population in equilibrium in random emitting population, then definitely the frequency should be 1. Wherever you're getting 1, you just choose that option. Then, where that question? For example, if the predominantly self-fertilizing plant regularly cross-pollinated with a frequency of 3%, assuming that no selection, what will be the percent of heterozygote at a locus with the two alleles showing gene frequencies 0.3 and 0.1? You just substitute this one 2pq 1 minus 1, 2 into pq into 1 minus fe. You will get this formula. Fe is because he's saying that it's a self-fertilizing and you are allowing for cross means you had because self-fertilizing means what happens? The only 3% is cross-pollinated means 97% is your inbreeding means you have to eliminate this one. Okay, means 0.97 percentage is your inbreeding. We have to eliminate this compound. That's why we have to eliminate this compound 1 minus 0.e. Then how to estimate this one? S by 2 minus s. Your s is your percent of your cross pollination. 0.097 divided by 2 minus 0.97. Okay, you will get one value. Then you substitute this Fe value in this formula 2pq 1 minus a. He asked percent of heterozygotes when there is a 3% of cross pollination. You just substitute this one, you will get the answer. Then if a gene M has 10 alleles and M1 to M10, how many number of genotypes? It's simple. You just substitute n into n plus 1 divided by 2, you will get this result. Then what is the formula of SD root by n? It is just standard error. Then by this I completed except biometry of heterosis. I covered almost all the topics of quantitative genetics. Okay, unfortunately no one is present. I expected a lot of doubts and lot of clarifications. Okay, but no one is there. But I finished most of the quantitative genetics chapters. If you have any doubts, you can ask in the group or you can ask now. I think only one participant is there. K, I don't know who is K. You are there?
Yes,
Hello.
sir.
Any doubts? I'm gone
No,
very fastly, okay? Because you also not questioned anywhere. Any doubts? I
yes,
know many doubts will be there. It's very difficult. Nobody stopped
yes,
me. Yeah, yeah. Because it's very difficult. If somebody is there to ask the doubts, because if you not wherever you not understood, you ask now. I will better. I will explain so that everyone will get.
okay, sir.
Any doubts
Sir,
in
not
any?
still. I will go through once
Okay, okay, okay. I know, I
book.
know. It's very difficult. I went through
Yes,
very
sir.
fast. Yeah,
Yes, sir.
because
That graph also, sir. G into E interactions.
oh, actually, it's very difficult. Actually, I went through because now I was reading continuously because I was understood now. Again,
Yes,
from telling from the basics, very difficult. Somebody
yes,
is
sir.
there to interact. I will ask the
Yes, sir.
question to someone. Somebody will ask again the doubt. It's like kind of interaction. It will be better. If I was teaching
Yes,
alone, it will go fluent, like with the flow. Okay.
yes,
If
sir.
any doubts,
Yes.
please don't hesitate to ask anytime. Okay.
Okay, sir. Okay, sir.
Bye bye. I completed almost all the questions. You understood this how to solve this one? I
Yes,
given
sir.
the
Numericals.
formulas. Yeah, you just substitute in that one. You will get the answer. Okay. Bye bye.
Sir, in numerical sections, only this type of question will be asked now.
Yeah, yeah, because there is no because see in like 50 will be there na 50 questions. Like see
Yes, sir.
out of 50 questions in last year, 14 questions were asked from this section. That's why I
Okay, sir.
prepared 200 slides. Okay.
Okay, sir. From this
See,
chapter also, sir. G into interaction.
yeah, yeah. See this one. This AMI model. See this model I have explained now. This was
Yes,
from
sir.
G interaction only. AMI and
O-
biplot model. Okay. And see all the questions are from 2024 paper. The how many number of
okay, sir.
questions they were asked in a single
Oh,
paper. This many weightage was nearly 13 to 12 questions, 13 to 14 questions from the quantitative genetics.
oh,
Okay. That's why it's having a very high weightage. I thought to discuss in detail. No one is there. Okay. It's
oh,
very, very
yes, sir.
important, and it's very, very important to read. See, I have covered from multiple sources. Uh, Majorly, you read J.R. Sharma book for GE interaction, and you read
Yes, sir.
one book called as I was keeping in my table, and I will on the video, and I will show. You can refer this book called as um, Quantitative Genetics by uh, I think it is Nandarajan and Manivannan Gunashekaran. This book, or you can refer J.R. Sharma book, because
Yes,
I
sir.
made the notes actually. I made the notes from all the books, quantity genetics. That's why I was giving able to make the presentation. Okay, read from this
Mm.
book. If you not understand J.R. Sharma book, read this book. It's very simple book, and all the for every analysis he given the case studies, numerical problems. Like, but it is unnecessary to solve here because it takes a lot of time. Normally, I do all the analysis in R. If anybody want to analyze your data, you can contact me. Okay. I recently
Okay,
I
sir.
did line tester for my one of the junior, and you see that it is not that much very difficult that what you are studying. There are one package called as plant breeding package. In
Yes,
R,
sir.
in our studio, we having one breeding package called as plant breeding package. Just
Mm.
you give your upload your data, just it will give all functions are preloaded. You have to specify your data and it will give results in a minute. No need to read all those things. Okay. And plant breeding package. There is another package called as Agricola. To do all your ANOVA, either in which designs, augmented, RCBD, whatever design, just use this Agricola package. Then you can get all the analysis in a single go. And
Okay,
but
sir.
what we need is we need the basic knowledge about our studio, how to use the commands, how to use the codes. You should know that much. Okay,
Yes, sir.
bye. Okay, good night. If any doubts, you can ask. Bye.
Okay, sir. Thank you, sir.
Bye. Bye. Thank you for patient listening. Good night. Good night.
Okay, sir. Thank you, sir.